Now, perhaps you don't know some of the terminologies that I've been using so far in describing this Baltimore skein, so some definitions are in order. A messenger RNA, or mRNA, which is ready to be translated, has been assigned the plus strand polarity. This has nothing to do with electric charge or anything else. It's just a convention. mRNA is called the plus strand because as many nucleic acids, there are two strands, so we have to designate one or the other. DNA of the equivalent polarity as mRNA is also called the plus strand. Now, DNA is not translated. This is just a convention to show that this strand of DNA is the same polarity as mRNA. Both RNA and DNA complements of plus strands are called minus or negative strands. And finally, remember, not all plus-stranded RNA is mRNA. Just because an RNA is of positive polarity doesn't mean it's going to be translated. On the other hand, if we call it mRNA, that means it will be translated. Let's take a little bit of a look at the viral genome now. We've spent some time talking about how it was discovered and how it can be categorized. What's encoded in a viral genome? Well, we know that there are gene products and regulatory signals to replicate the genome. Viruses need to reproduce, so the genome has to be replicated. We know that there are signals for putting together the genome into a virus particle. We call that assembly and packaging. We know there are genes for regulating and timing the replication cycle. When a virus enters a cell, not everything happens at once. It would not make sense, for example, to produce structural proteins until they're needed for assembly of new virus particles. So that timing is encoded in the genome. Many viruses encode at least one protein that modulates host defenses. If they don't, they would no longer exist on the planet. And finally, there are genes encoding proteins that are needed to spread from that first cell that's infected to other cells or other hosts. And these are just some of the gene products encoded in a genome. We do know what is not encoded in a genome. There are no genes encoding a complete protein synthesis machinery. That's why viral mRNA has to be ready for host cell translation. Some viral genomes do contain genes that encode components of the translation system, like amino-acyl tRNA synthetases, initiation factors, and even transfer RNAs. But no virus encodes the complete translation system. There are no genes encoding proteins involved in energy production or membrane biosynthesis. Again, those have to come from the cell. And there are no classical centromeres or telomeres that you would find in a standard host chromosome. The absence of such genes, as well as many others, is why viruses are obligate intracellular parasites. Now, it may be that we simply haven't discovered some of these genes. We discover viruses on a daily basis, hundreds and hundreds every day, and over half of the genes that are in these new viruses we've never seen before. So it could be that some of the functions that we just said are not in genomes will eventually be found to be there as well. Let's take a look at some of the extremes of viruses, the biggest and the smallest known virus genomes, just to give you an idea of how many nucleotides and proteins are in these. The biggest virus genome identified to date is from a virus called Pandoravirus salinus. It's over 2.4 million base pairs of DNA long, and it encodes 2,541 proteins. This is absolutely huge. The average virus is far, far smaller than this. And this goes down a list of genomes to what is a relatively small genome of 610,000 for Pithovirus sibiricum, encoding 467 proteins. So these are the top 10 or 12 genomes according to size, ranging from 2.5 million down to 600,000 base pairs. These are really the giants in the virus world. At the other extreme are the small virus genomes. Let's take a look at some of those. The smallest known viral genome is from an agent called a viroid. It's 120 nucleotides of circular RNA, and it encodes no proteins. These are typically plant pathogens that enter plant cells and cause disease without encoding any proteins. Really, really remarkable. And again, a slightly larger genome, 4,000 bases in length, encoding 7 proteins. And in between these extremes, you can see there are some viruses that encode just 1 or 2 proteins. So truly the virus world is remarkable. There's a huge range, and it's quite different from, say, Homo sapiens, where our genomes are more or less the same length.